Can you see the screen?
Yes, it's very clear.
Okay.
Yeah.
Well, thank you, Samuel, for inviting me. It was a bit of a random encounter, right? We met about a month ago at the SusMat in Singapore. We're just sitting next to each other. It was a very pleasant conversation. And then you just asked me to give a talk, and it was just, that's what conferences are for. So thank you. And so I'll go straight to the topic of my talk on this peptide coacervate for intracellular delivery of macromolecule. And I give a bit of a background, and hopefully I can stay within the half an hour. But if I'm spending too much time at some point, please just let me know in case I'm losing track of time. So for many therapeutics, or promising therapeutic, we want to be able to introduce large therapeutic inside cells, right? So that could include, for instance, protein therapeutic like antibody, gene editing like CRISPR-Cas, mRNA, of course, we all heard about mRNA-based therapy during the COVID. But of course, the cell is not meant to have foreign body entering the cell. So there is this billion-year-old barrier that is there to prevent things from entering inside cells and infecting cells. So there's enormous interest in being able to deliver large therapeutic inside cells. There's different solution at the moment. I won't go through all of them, but I can maybe give a short background. It's not meant to be exhaustive, but this is probably the main one. Of course, everyone heard about the lipid nanoparticle because this is what was used during the COVID. But there is some problem there. The formulation tends to be complex. It's also very lengthy. There is some cytotoxicity. There's also some issue with the IP. Actually, it's very prohibitive, and it's difficult to access actually. For people who come from the biology or molecular biology, oftentimes they may use those viral vector, but there's some other issue with this, for instance, immunogenic toxicity. And in fact, there's a strong shift towards going towards non-viral vector to deliver large macromolecule inside cells. This is more the area of material scientists, right? Like polymer nanoparticle. And a lot of people who come from polymer background have started developing all sort of small nanoparticle made of polymer. But as I will explain later, you need to escape this endosome, right? Escaping this those compartment once it's inside the cell. There's also some other issue. There's a few more system that exists. I won't go through all of them. And what I'll explain instead is what we've developed in the past few years. And the story I'd like to tell is how actually this started from complete fundamental research, and we had no idea that it would end up into something that can be used for therapeutic. And so the story I'd like to tell today is how very basic studies and science into this pretty aggressive animal, the giant squid, led into new therapeutic for gene delivery, gene editing, and other kind of therapy. So I won't have time today to explain everything, but this started many years ago when I was a postdoc in California. And we were interested. I come from a mechanics of materials background, so we were interested by this animal. It's a pretty large animal, but human size. And then inside the mouth, there is this thing called the beak, the squid beak, and it's actually very hard and very strong. And what we discovered at the time is that actually this beak here is not calcified. It's completely made of organic molecules. So it's mostly protein and polysaccharide complex together in a very complex way with a graded structure. It's a long story. So it started with this squid beak, and then we were mostly interested at first into the mechanics of that material. Now, if you want to do what I call molecular biomimetics, you need to understand what is the component of those structure, right? And we had no idea at the time. So So if I think about this molecular biomimicry, the grand challenge, like this is some model system people have studied over the years, for instance, seashell, muscle adhesive, silk, right? You can find anywhere in any of your backyard or near the sea, wherever you live, some of those animals. And in fact, I would argue any country may have some interesting system to study. So we've made, very good progress in the past decades in understanding the composition of those structures. For instance, could be made of protein, and we're able to sequence them. I'll come back to this in a second. They're also made of polysaccharide and biominerals, and there's all sort of very sophisticated tool to study them. And then, you can, for instance, study their mechanical performance, maybe their optical property, etc. But what is, in my view, still a bit missing and difficult is, here is how does nature go from the building block to the final structure? And if we want to mimic natural materials that you find in nature that are really phenomenal and have been optimized over millions of years of evolution, you need to understand this so-called biofabrication process. And this is actually not that simple, and that's where, in my view, there's a gap. Okay. So we started on this journey more than 12 years ago, 13 years ago, a little bit after I moved from the US to Singapore. And this approach that we've adopted is basically to integrate, at the time, the latest tools in sequencing technology and proteomic that we combine with material science. So you may have your system of interest. Let me put my laser here, it'll be easier here. Okay, here we go. For instance, this is the model natural system of interest. You can extract the cell or the tissue that will form the final materials of interest. You do RNA sequencing. At the time, we were the first one to do that for biomaterials. It was 12 years ago, or even a bit more. You also do proteomics, material science, and long story short, you can identify complete new genes that are producing the final materials of interest. Okay. So that's the overall story. And if you have an interest to understand more, you can find some detail, for instance, in this review here. And if you don't have access to it, at the end of the talk, or send you an email, I'd be happy to send you the paper. Okay, so at the time, so we were studying this very interesting and intriguing, very hard and strong beak of those of this squid. In fact, any squid have some of those. And so we basically extracted cell from, it's called the baculoblast. Those are the cells that form the final tissue. And long story short, we also am able to extract some peptide from this beak because it's mostly made of protein. And then we were able to identify the protein that are basically the main elements of this beak. Of course, they're highly cross-linked. It's like a very highly densely cross-linked polymer. But we find those protein, and then we started looking at them, and we discovered some interesting feature there. So if you look at the sequence, I don't know if people here have some background in biochemistry, but if you look at the protein sequence, you see this kind of polymer block copolymer architecture. Sorry, those light blue and dark blue region. They're very rich. This is the amino acid sequence. So they're rich in glycine, this other amino acid histidine, sorry. And then when we look at what it looks like, this pattern, it looks very similar to this other peptide called elastin-like polypeptide, right? Elastin is the main protein in human skin together with collagen. And so what we know with this peptide is that they can form this so-called phase separation. So I think a video will be more telling of what's happening. So what I'm showing you here, I'll show you in a second, is some small peptide that we derived from this protein. And initially you put it in acetic acid, so it's soluble, it's pH 3 or 4, it's basically vinegar. And that will be in the pipette. Okay, so you see the pipette here. Then you pipette this inside the buffer that has pH 7, neutral pH. And spontaneously you see you have this kind of cloudy phase forming. Okay. you can then disperse it, and it looks something like this. And then if you're a biochemist or molecular biologist, your first impression is to think, well, this is a bit boring, maybe there's aggregation. But in fact, if you look at this under the microscope, just a simple optical microscope, what you see is this, you see those little droplets, okay? So you have those concentrated droplets, I'll come this to in a second, that's spontaneous. So that's why you have a cloudy phase, because now you have light scattering from those microscale droplets. Why the animal is using this strategy is a complete whole different story. I won't have time to explain it today, but I want to explain to you or describe to you what we're using with those droplets now. So before going this, so what we just witnessed here is called liquid-liquid phase separation, LLPS, which is also called coacervation. And actually, this is a mechanism that has been known for decades in the area of colloidal chemistry and material science of complex fluid, right? So basically, you imagine this is a phase diagram where you may have a homogeneous solution of polymer or biopolymer, in that case, a peptide. And then when you change maybe the temperature or the pH, in that case, where it's just so, or ionic strength maybe, then you have phase separation happening. You have two phases. You have one which is concentrated in the polymer, and then you have a dilute phase around it, okay? And so those droplets are called coacervate or condensate by the biologists, and there's a high concentration of the polymer inside. And this is a simple phase diagram. And if you're a material scientist, you might be more familiar with this kind of phase diagram here, where you can see maybe the binodal, the spinodal. And this is basically the same principle here. All right, but what is key, and let me go back to a second here, is when I show you this video here, if you have any macromolecule that is inside this in solution here, somehow when you do this process of phase separation, a lot of them will actually get entrapped inside those droplets with very high efficiency, like up to 95%. And this happens in a few seconds, it's spontaneous. So it means you can encapsulate all sort of therapeutic, all sort of macromolecule, including therapeutic inside the droplet. So this process of coacervation, it's very interesting for therapeutic because the whole process is done under aqueous condition. So there's no organic solvent. You have very high encapsulation efficiency, up to 90 or over 90%. The droplet, they're kind of still liquid-like, okay? And then you can also recruit different modality, and most importantly, you can make them stimular responsive. So what I mean here is that you can disassemble those coacervates either endogenically by encoding something inside the peptide, or by external trigger, for instance, a magnetic field or near-infrared, if you encapsulate other kind of inorganic nanoparticle inside the droplet. And so when we first studied this a few years ago, as a first proof of principle, we were interested in showing that therapeutic can be recruited within the droplet, and then the droplet can be disassembled by an external stimuli. That was kind of, we just wanted to see, can we do that? And so we started by encapsulating insulin and glucose oxidase inside those droplets spontaneously, as I showed you earlier. And then when you flow glucose, because you have glucose oxidase here, it will become gluconic acid. So it will acidify the inside of this droplet. And when it's acidic, the droplet disassemble and release the insulin on demand, basically according to glucose level. So that was just a proof of principle. And all what I'm showing you here is that you have the insulin, which is labeled, green labeled, inside the droplet is green. You have glucose oxidase, which is red label in red, and we put them together, the yellow. And so I don't have all the data here, I don't have time, but basically when we did this, you flow glucose and then you disassemble the droplet and you release the insulin on demand. We never ever did any in vivo study or anything, it was just a proof of principle. But when we were working on this, we found something very interesting. We found that we're doing my student was doing some cell culture, and he found those droplets inside the cell, which is very intriguing because if anyone works on drug delivery here, usually the consensus is that you have to have vectors that are below 200 nanometers to deliver inside cells. But those droplets are 1 to 2 micron. So it's very strange. So they stay inside the cell, but they wouldn't disassemble. So if you want to deliver something in a therapeutic inside cells, it's great that you can go inside the cell, of course. But if they don't disassemble and release, it's useless. And so this is where I got a very, very smart and clever student that joined my lab at the time, Sunyu here. And I think I'll go a bit fast because otherwise I won't have time. So basically what he did, he modified slightly the chemistry of this peptide. He added one charge residue, here it's a lysine. And then to this, he actually modified further with this chemistry here. This is called self-immolative chemistry together disulfide bond. So let me try to explain in a nutshell what's happening here. So if you don't have this modification, if you take the original peptide, what you see here is the turbidity, okay? If you high turbidity, it means you have coacervation. If you below here, you mean you're in solution. So if you look at the original peptides called HB-PepK at pH 758 or something, there is no coacervation. But once you do the chemical modification, you see that at pH 6 or 7 here, you actually have this phase separation. So spontaneously you form this phase separation. But then if this is, if you flow, or once it goes inside the cell, those droplets will be subjected to this peptide called glutathione, which is highly upregulated inside cells, especially cancer cell. And what this glutathione does is that it cleaves this disulfide bond, and then you have this self-immolative chemistry. So the whole side chain is being removed and cleaved. And so you're back to the initial peptide, but now you are also pH 6 or something. So it means that the peptide inside the cell will spontaneously disassemble and release the cargo. If you didn't follow, there is a cartoon which should be easier to understand here. So basically you have this initial peptide with an extra charge that we add, it's a lysine. You modify, and then under those conditions, you don't have phase separation. Then this peptide is chemically modified. So you actually block this charge, you're able to form those coacervate. And if you induce this coacervation in the presence of therapeutic, which are in solution, then the therapeutic will get entrapped inside the droplet. And then this droplet that is shown here enter the cell through a process which is not through the classical way that they usually goes inside the cell. It get reduced, and then spontaneously it disassemble, and it deliver the payload. And so it's very powerful because any macromolecule in solution here, as long as it goes inside the droplet, it can be delivered. So what we've shown, for instance, is that inside cell, we could deliver all sort of protein up to like 430 kilodaltons an enzyme. And then we maintain the activity, you can deliver enzyme, antibody, and all of this was verified. So it was very powerful. Now those are proof of concept of like model system, but can you actually deliver actual therapeutic? So this is some example here. So in the just quick background. So this is some work with Karolinska Institute in Sweden. In that case, the target is this undruggable oncogene called MYC. And MYC is a transcription factor. It's one of the most commonly amplified oncogene in cancer. So it's actually deregulated in 70% of human cancer. I'm not a cancer scientist, but basically the idea is that this oncogene is very difficult to target. But a few years ago, some group in Spain have come up with this peptide called OMA-MYC, which can in principle target this oncogene and prevent this dysregulation. But the problem is that this peptide has very poor cellular penetration. It tends to aggregate, and there's also some reproducibility in the literature. So the idea was, can we actually deliver this very promising peptide with our system? And so the answer is yes. You see some cell here that if you try to, so the peptide here, the therapeutic is labeled, with a green label, and you can see that if you try to deliver by itself, nothing happens. You don't see it inside the cell, and then once you put it inside our droplets, inside the coacervate, they actually go very nicely inside the cell because the cells are green. I won't spend too much time here, but basically this is all sort of cell study. And then what was shown here is that if you look at the cell viability of those cells here, that you can see that you have basically very low cell viability. So the peptide goes inside the cell with our system and can kill the cell. And then here you have some marker of apoptosis, so cell death. I won't spend too much more time, but the message here is that we have those peptides that were thought to be difficult to deliver inside cells, those therapeutic peptides, but with our system, we can deliver them. And then in the rest of the talk, I'll show you what other kind of therapeutic we can deliver inside cells. So in that case here, for instance, this is a mRNA. So this was started before COVID. It was a collaborator from a company who told me that the future is mRNA, you have to deliver mRNA. And so long story short, it worked very well. So here, this is mRNA encoding for luciferase, and this is a comparison with the lipofectamine, which is kind of the gold standard. This is the first generation. The next generation actually works even better. This is mRNA encoding for green fluorescent protein. You can see everything is green. And then based on the fax data, you actually have basically up to more than 90% of cells that uptake the coacervate, and you have 82% of expression. So it works very well. This is actually with a pharma company. This is in vitro. And then what you can see here, this is the activity. This is log scale here. And what you see is the traditional lipid nanoparticle here. So actually in vitro, that works actually better. So we have a new delivery system that can outperform the kind of the best-in-class lipid nanoparticle at the moment. Of course, the question here is, how do those strange coacervate droplets enter the cell? And then at first we had no idea, but by using this special scanning TM microscopy technique, we were able to identify that those droplets are being uptaken in the cell by mechanisms that share features of both macropinocytosis and phagocytosis. What you see here are cells. This is a EM-TM section, and this is the droplet, that very large here, and you can see basically that it's being basically grabbed by so-called filipodia. Those are cell structure that basically made of actin that capture the droplet and then pull them inside the cell. This is an SCM image, and the same image, if you do a false coloring, it looks something like this. So you can see basically here, this is the droplets. Together inside you have the therapeutic, and then you have those filipodia here, those protrusion from the cell membrane who comes out, grab those droplets and bring them in, and then it will disassemble what's inside the cell and deliver. I'll go faster here because we don't have time. And I just want to show you that now that we have those coacervate made of fairly simple peptide, they're only 26 amino acid long, you can actually modify the sequence of the peptide and then improve further the uptake. Okay, so basically this is the sequence of a peptide, right? The amino acid sequence. And then you keep most of the amino acid residue the same, but then you change some in very specific location, you do some specific mutation. And then we make those peptides by solid state synthesis. And so, for instance, you may have some aromatic residue in this fourth position or charge, etc. So we can just change 2 or 3 amino acids. And what we found is that this changed completely the mechanical property or the material property of those droplets. And some of them become more liquid-like, or some of them are more gel-like by just a few amino acid mutation. I will skip this. Here, I don't think I have time to explain everything, but what I want to mention is that depending on the amino acid residue that we add inside our peptide, we can make those droplets more kind of gel-like, so a bit stiffer, or liquid-like, where they really maintain their liquid-like behavior, okay? And the reason I'm saying you this, I'll go a bit faster here, is that if it's more liquid-like, it's based on molecular dynamic simulation that we do with our colleague here in Singapore. what we found is that it's because you have more molecular hydration. So you have hydrogen bonded molecule in specific region of the peptide. But let me explain a bit faster here. Okay, so what I want to show you here now is the delivery of different kind of therapeutic with a different kind of peptide. So you can focus on the top here, for instance. So what you see here is the different peptide. This is the original one. This is the new one, new version. And then this is here we deliver a green fluorescent protein inside cells. And by just changing the sequence of those peptides, just those 2 or 3 mutations, you have very different efficacy, right? So what you see here, for instance, in red, this is the fluorescence intensity that from the FACS sorting that they use in cell biology. And then you can see that you have a much higher efficacy in some of those peptides by just changing a few amino acids. And the reason is because it's a bit more liquid-like and it has more bonding to the cell membrane. And so we can deliver very efficiently. In that case, this is a green fluorescent protein. This is an antibody. This is a red fluorescent protein. This is another larger enzyme. So it works really well. And then we have really improved the system here. Saporin is a cytotoxic protein. It's kind of a reporter. So if you have strong cytotoxicity, it means that this toxic protein is really efficiently released inside the cell. So if you look at the best peptide here in orange, you have basically almost 100% cell death, which means it works very well. So we are very excited about this. We can also deliver a gene, mRNA. We can also tune the efficacy. I'll go a bit faster here. And what can also be done, and I'll finish in the last few minutes here, you can also deliver modality that I use for gene editing, so CRISPR-Cas9. So if you want to deliver CRISPR-Cas9 inside a cell, there's 3 options. You can deliver the entire gene that encodes for both the enzyme and the signal guide RNA. You can deliver the complex of messenger RNA and guide RNA, or you can deliver what is called the RNP complex. So all 3, usually, if you want to do it in the lab, you need to buy transfection reagents that are very expensive. But in our case, you can use one system only, and you can deliver all 3 if you want to system. And then what we've demonstrated is that we actually managed to deliver inside cell this gene editing modality, and we can get pretty high efficiency of gene editing. So as an example here, for instance, this is this HeLa cell that expresses green fluorescent protein. And then we knock out the gene with the Cas9. You can see here that once you knock it out, you don't see almost no green anymore because it's been knocked down. And then maybe I'll finish with this. When we start talking to a colleague who works, for instance, with different kind of cell, they say, well, this is great, but can you transfect? Very difficult to transfect cells. For instance, primary cells are notoriously difficult to transfect. So here, this is delivery of green fluorescent protein in this cell. You can see everything is nicely green. This is mRNA encoding for GFP. T cells, so T cells are used for immunotherapy. And then you can see that you managed to transfer them. We're very excited by this. And also macrophage. And macrophage is actually, it's also an interesting one because the biologists who work in this field, it's well known that it's difficult to do transfection. And so here we managed to, with our system, to deliver protein, mRNA with very high efficacy. You can see the red bars here. So it works very well. And so you can start thinking about using our platform for immunotherapy, for instance. I think in the interest of time, I'll skip the last 2 slides, but I'll go to the conclusion instead. And so what I want to explain today that I just showed you is that, so we've developed in the past few years, this new delivery system called stimular responsive peptide coacervate. And it's basically a universal delivery vehicle to deliver any kind of macromolecule inside cells. So you can deliver antibody, enzyme, therapeutic peptide, nucleic acid. So there's a huge potential for all sort of therapeutic application. I went a bit fast, and I apologize for that because I didn't want to spend too much time. But basically, you can modulate the material property, right? After all, this is a material science audience here. by just changing a few amino acids in the peptide sequence, and you can make those droplets more gel-like or more liquid-like. And depending on those properties, you regulate how it binds to the cell membrane, and you also regulate the uptake and eventually the release kinetic of the cargo, which is very important for different kind of therapeutic application. And there's some other stuff. And then the last thing I'd like to say is that everything started here from complete blue sky, curiosity-driven research. When we started this program, we had no idea we'd be working in drug delivery, but it turns out that by just being curious, you can see some very interesting things. So started from this animal here. This is the giant squid. I got entangled inside a tentacle here. You can see my face here. And so that's how it all started. Okay, and there's a lot of people who have worked over the years on that project. The people in red here are the main people that I showed today, and then this is all the collaborator that we work on this topic. And then we're very fortunate in Singapore to have very good funding for scientific research. So with this, I hope I didn't spend too much time. So there's still some time for question. So happy to answer any question you may have.
Thank you very much, Professor Miserez, for that insightful talk. I wish we had more time, so at least you could have expanded more. But
Yeah, I know.
yeah, but I'm sure I think people have picked up all the exciting things from here. So we can now ask questions or make comments. You can either raise your hand or just unmute yourself and ask a question. Anyone, or you can type in the chat. Okay, so I think while people are still thinking about their questions, probably I can just ask one question.
I was wondering maybe a lot of people don't have biology or bio background. That could be the challenge here. I don't know if people come from biomaterials here or not.
No, I know some people that have got the background.
Oh, okay, well, don't be shy then.
No, they're going to ask. It's coming through because I can see. I think that's Professor Kuma from FIT's Advanced Drug Delivery Platform. I'm sure he's going to ask some questions there. And also there's Dr. Manseka. He's also going to ask. I know they'll come through.
Okay.
Yeah. So I think maybe for me the question that I want to ask is just the part where you talk about controlling the droplet from being gel-like to the being liquid-like. What are the
Yes.
key parameters that you optimize to be able to have total
Yeah.
control of that? The reason why I'm asking that is that I think you also mentioned in your conclusion that in addition to the specific application that you're talking about in this case,
Yes.
you also look at
So
expanding to go to biosynthesis and energy.
yeah. So I went a bit. Do you still see my screen? It's still shared, right?
Yeah, it's showing a third generation of
Okay.
peptides. Yeah.
So what we've done here, I went a bit fast. I apologize for that. This is the peptide sequence, and you can see that basically we keep this. There's a penta repeat G, so glycine, histidine, glycine, and X can be whatever we want, and Y is tyrosine. So this fourth residue is changed systematically. So if you put something which is more hydrophobic here, okay, like valine or alanine, then it's more, the eventual peptide will be more gel-like. If you put more charged amino acids, okay, for instance, arginine here, this is our arginine, or maybe aspartic acid, then it becomes more liquid-like because you have charged. And those charge here, those residue, they can actually bond through hydrogen bond a water molecule. So basically, you just have to change the whole gel-like versus liquid-like is controlled by a few amino acid mutation where it's either more hydrophobic or hydrophilic. And when it's more hydrophilic, what we know from the simulation is that here you have basically more residue that are hydrogen bonded. Okay, you can see here in red here. So there's so this is basically just having a few amino acids that are more hydrophilic. That's how you control the property of more more gel or liquid-like. Does he answer your question?
Yeah, no, yeah, thank you very much. So in terms of, I think you talked about moving to biosensing and energy applications.
Yeah,
Uh,
this is something it's ongoing. We
okay,
currently working on the manuscript, so it's not there yet. It's kind of
okay,
exciting. I cannot share too much yet because we haven't published yet.
okay, I understand. No, thank you very much for that. Okay, I'll just say, okay, before let me just see the questions here.
So some people had
Yeah,
some questions
I think
already.
there was hand that was raised, but I'll just start off by reading the questions here. I think the first question is from Professor Kuma.
Yeah,
He said, I can relate to, I don't know, can you see from there? I
yeah,
can,
yeah. I
rel
can see.
yeah, he
Yeah,
said
yeah,
I can
yeah.
relate, yeah, yeah.
Yeah, yeah.
I, yeah, I can relate to elastin very well. The question I provided while registering was how scalable these peptides are. I don't know, is
Yeah.
that question clear enough, or should I ask him to talk through?
Yeah, so actually, we make those by solid state peptide synthesis. So as long as these days you have those large scale peptide synthesizers, so it's not really an issue to scale it up. And the question about elastin, indeed, in fact, when we first look at this, we saw some similarity with elastin and tropoelastin. So that's where we had the idea of trying to see if we can also have a coacervate like tropoelastin. I know very well the work of my friend Tosh Chilcotti at Duke. So it kind of came from that. So we can scale it. That shouldn't be an issue. At the moment, we do mostly the research, but we're trying to launch a startup, and so that question will become very important.
Okay, thank you. Professor Kuma, is your question answered, or you want a follow-up comment? I'm not sure if we can talk.
Can you? I don't know if anyone are you able to unmute, or maybe
You
you
should be
have the
able to.
control over everything, no?
Let me just check. He's supposed to because everyone is it's a meeting, set up as a meeting.
Oh, okay. He's unmuted now.
Yeah. Okay. Anyway, let's go to the next question. That's from Nombeko Skosana. I think there are two. Have you started working on functionalizing?
Yes.
Okay. And then for example,
That's
targeting spaces.
yeah, that's an excellent question. Yes, the beauty of this system, it's fairly easy to incorporate other kind of molecule inside the droplet just when you form the phase separation. So you can actually incorporate ligand inside a peptide. This is something we're currently working on. So of course, targeted drug gene delivery is the holy grail in this field, right? Like that person must realize. So that's something we currently working on, and we have quite promising data actually. And then the second question is if you have stability tests and if they maintain their integrity. So it's interesting because the stability, it all depends on the definition. And so they tend to stand that some of them will kind of grow over time a little bit, but it turns out that whether they're, let's say, 800 nanometers or 1 or even 2 micron, they always get uptaken inside the cell. But that being said, we have some new formulation now that remain stable in terms of size for a long time. And then as long as we don't disassemble them, the um, the droplet, they will maintain, the encapsulated cargo will remain inside. This we have verified. Of course, if you change the pH, for instance, but this would happen only once it's inside the cell, they will get disassembled. So, I mean, it seems it's working quite well in that sense, but there is still improvement to be done, of course.
Okay, no, thank you. Nombeko, do you have any follow-up comment on that?
Yeah, just maybe one more. Regarding the size of the microdroplets, is it possible to control the sizes, or the sizes vary?
Yes, so in the latest generation, we just published this. We managed to control them from like 100 nanometers to like 1 micron. But there's something very interesting because I don't know if you work in drug delivery, but usually people always think that, oh, it has to be 200 nanometers, otherwise it's not very good. It turns out, especially for immune cell, that the large droplet work actually much better than the smaller one. And the reason we think it's not fully verified is because it doesn't go through the classical endocytic pathway. So if you go through the classical endocytic pathway, you end up in the endosome, right, where you get degraded. But because our system goes through a different pathway, through this macropinocytosis, it actually doesn't get degraded inside an endosome-like compartment inside the cell. And also it goes inside what is called the macropinosome, which is actually easier to disassemble. And therefore you kind of avoid this whole degradation problem that a lot of people are struggling with LNP.
Mm. Oh yeah, and also if that's the case, then it's easier to actually encapsulate larger genetic material, because I
Yeah.
know that in gene therapy, that's the main challenge right now.
Yes, so
Having
I can tell you, I cannot disclose all of it because we have some confidentiality agreement. But so protein, we've been able to encapsulate and deliver enzyme up to 470 kilodalton, which is enormous. And in terms of genes, lately we've managed to deliver genes, plasmid as large as 12 kilobase and mRNA as large as 8 kilobase. And I don't know if you work in this field, but I think if you do, you appreciate that this is actually very challenging with traditional method.
Mm. Yeah,
So
that's true.
we managed to do it at the moment.
Okay, that's awesome.
Thank
Thanks.
you.
I can see the comment from Professor Kuma. I think he looks like he's not able to talk.
Yeah, I see that. Yeah.
Yeah, indeed. And then there is a comment by Professor Bala.
Yeah.
He said he's interested more in comments on emerging applications. I know that's all you say. You can't really say much about it,
Well,
but
I can tell you something. It's very, okay, so it's very fundamental study at the moment, but the key is that, and then other people are working on this, is that we can actually, uh, inside a droplet, you have a very high concentration of whatever you incorporate, and therefore the activity, the chemical activity is very high. So for instance, if you put an enzyme inside a droplet, the activity goes through the roof. And so that's kind of the idea is you can put kind of enzyme or nanozyme inside those droplets and you can really accelerate any kind of process inside the droplet. It's very basic science at the moment, but we have the data look good in the lab. And so that's what I mean that there's some interesting potential in the energy sector. But we know that the level where we can start to, translate the technology, unlike the drug delivery. I hope that answered your question.
Professor Bala, you have any follow-up comments?
Hello, everyone. Just wanted to say thank you, Prof, for the exciting talk and also for the very, very fundamental work that is full of science you are busy in sharing with our African community. Yes, indeed. I was just looking on the side of the wettability of this, in which I believe it is really interesting when for the next generation of I could say any storage devices like sodium-ion batteries and so on.
Yeah.
So yeah, that was a little bit, but I'm okay with the comment. Thank you very much. Thank you, Sam.
Thank you for attending. And then I just want to mention if some of the audience doesn't have access to some of the publication, I'd be happy to share them, because sometimes it's not always open access. So you ask me, I'll send, or I can share with Samuel and Samuel can share with other people. In case you don't have access, just let me know.
Okay, yeah. Thank you very much for that. I think that is great because I think the whole idea of the webinars is that it should stimulate our thinking, and then we can see what work we can then explore beyond this.
Sure.
So I think maybe for the benefit of the audience, in terms of opportunities for doing research work together, I think you mentioned the funding. Is there maybe specific funding that can support international maybe postdocs or something along those lines?
So what there is in Singapore, but I think they're changing the scheme. But for more than 15 years, they had something for PhD student called the Singa Scholarship. And so this Singa Scholarship is basically trying to increase the diversity of researcher in Singapore beyond what they call traditional researcher here in Singapore, which will be a student from China or India. And so over the years, they had this Singa opportunity for students from around the world So in my lab, for instance, over the years, I've had students from like Brazil, Europe, that's considered non-traditional here in Singapore. There have been students from Africa in some of the lab, not many, to be honest, but there are some opportunities. So if any talented PhD student is interested, they should look up this scheme called the Singa Scholarship, SINGA, because I know that they're trying to recruit students from different countries. Like I have students from Iran at the moment through this scheme. I went from Brazil. I've never had from Africa. I had a no, I haven't had it, but I've seen some of the students. So if there is a student here, or if some PI have motivated student, they should direct them to this Singa, and then that's open for any to work in any institution in Singapore. But this is specifically for PhD scholarship.
Okay, thank you. And then in terms of, because sometimes we get mobility grants in Africa, is that something that can be considered in such a way that maybe one can get funding to be able to maybe fly to Singapore and then spend some time, maybe a month or 3 months? For example, like if you look at TM, I know you got a very good cultural science facility.
Yeah. I need to ask.
Okay.
I'm not sure, to be honest.
Okay.
They may have, so because Singapore is always trying to kind of increase the talent pool beyond the immediate country. I have to check this. I know they have program like this for country in Southeast Asia, obviously, because this is in Southeast Asia. It's like, from, Vietnam, Indonesia, Thailand, but Africa, I'm not sure. I have to check.
Okay. Yeah. I'm not seeing any hand that is raised up by anyone or any comments in the chat. So I think probably I'll just maybe give the final comments. So I think maybe for the benefit of everyone, if you are not able to reach Professor Miserez, you can write to us and then I can facilitate the link. And then also beyond the meeting online, we are going to have our biennial conference in Nairobi in 2026. If Professor Miserez hasn't changed his mind, he had agreed to be with us.
When is it? Is it in December?
Yeah, December 2026.
Okay.
Next year.
If my schedule is free, I'd be happy to come. I always like coming to Africa. I haven't been in a long time, but if I don't have exam or teaching at this time, I'll try to make it.
Indeed, yeah, because I think that would be good to have the face-to-face interactions.
Sure.
Follow-up comments, I think that came through from the audience. So I think with that, I'll just hand over to Professor Bala. He's the president of the African MRS, to just close for us. Professor Bala.
Thank you very much, Dr. Samuel. Just to show my face to Prof. And for the volume
Where are
of
you
time,
based? So which country are you in?
You took to deliver for us this exciting, really, which is an emerging field of research in which I believe we don't have too much of African who are already involved on this type of research. I know people from WITS. I'm not sure if Kumur was here or in the meeting, but I know he's the most specialized person working on this area. So I think developing, as Samuel said, developing collaborations through the continent and also attending all our activity as like the upcoming African Material International Conference that we held each 2 years. It's really a platform in which most of Africans and international people are gathering really to develop collaboration and exchange some ideas. So we would like really to thank you for the time that you took to deliver this course, and also to text all attendees and telling that we must keep them updated that this webinar will start running again for each month, and we'll be able to really attract very interesting researchers around the world, around the continent, so that we can develop material science and engineering through the continent. So thank you, everyone. Thank you, Samuel, for organizing this. Merci beaucoup.
Thank you for having me. Where are you based? if I can just
I'm
ask.
based at the Université Cheikh Anta Diop in Sénégal, Dakar, in
Oh,
West
ok.
Africa.
Vous parlez
Yeah.
français?
Ah, bien sûr. Merci beaucoup.
Je suis
Merci.
francophone. Hein?
Ah, ça c'est bien, parfait. So Samuel will learn French then.
Merci beaucoup.
Okay, thank you very much.
Thank
Yeah,
you. I
thank
hope I see you in person. It will be really fun if I can make
Yes,
it.
indeed. Yes,
No,
indeed.
definitely.
Thank you.
Yeah,
Merci,
thank
au
you
revoir.
very much, everyone. Yeah,
Thank you,
all.
guys.
Thank you.
Bye.
Bye-bye.
Bye.